Introduction to Microbiology's Historical Journey. Welcome to this comprehensive overview of microbiology's history. Microbiology is the study of microorganisms, tiny living entities invisible to the naked eye. This field has transformed our understanding of disease, revolutionized medicine, and shaped modern science. Today, we'll explore how this discipline evolved from ancient observations to a sophisticated science that impacts every aspect of our lives. Join us as we uncover the fascinating history of how humans came to understand the microscopic world. Long before microbes were discovered, ancient civilizations developed theories about the causes and treatments of disease. Ancient Egyptians believed diseases were caused by divine punishment or blocked channels in the body that carried vital fluids. They treated illness with prayers to the gods and herbal remedies. The Greeks developed the influential theory of four bodily humors: blood, phlegm, black bile, and yellow bile. Disease was thought to occur when these humors were imbalanced. Romans expanded on Greek ideas while making practical advances in public health, building aqueducts and sewers based on the belief that clean water and sanitation could prevent disease. Hippocrates, who lived from 460 to 370 BCE, is known as the father of medicine. He rejected supernatural explanations for disease. Hippocrates proposed that illness resulted from natural factors like climate, diet, and living habits. He introduced the revolutionary concept of observing symptoms rationally and letting nature heal with minimal intervention. These early attempts to understand disease created the first systematic framework for medical practice and laid important groundwork for future scientific inquiry. Although these ancient theories were often incorrect by modern standards, they represented an important shift toward natural rather than supernatural explanations for disease. Some of these ideas, particularly the humoral theory, persisted in medical practice until the 19th century. The history of microbiology fundamentally changed with the invention of the microscope. In the late 16th century, Dutch spectacle makers Hans and Zacharias Janssen created the first compound microscope. Their innovative design combined multiple lenses to achieve greater magnification than any single lens could provide. Over the next several decades, various craftsmen made improvements to the microscope design. Robert Koch further developed this technology, creating more powerful and precise instruments in the 1660s. In 1665, Koch published Micrographia, the first major work devoted to microscopic observations. This groundbreaking publication revealed the microscopic world to scientific audiences for the first time. These technological advances provided the essential tool that would allow scientists to finally observe microorganisms directly. Antonie van Leeuwenhoek, a Dutch draper and scientist, lived from 1632 to 1723. His groundbreaking work would forever change our understanding of the microscopic world. Van Leeuwenhoek created powerful single-lens microscopes, achieving magnifications up to 270 times. His handcrafted instruments were far superior to others of his time, allowing him to see what no one had observed before. In 1676, van Leeuwenhoek observed what he called animalcules, tiny animals in water samples. This was the first documented observation of bacteria and protozoa, organisms no human had ever seen before. Van Leeuwenhoek documented his discoveries in detailed letters to the Royal Society of London. These contained meticulous observations and drawings of what he saw. Though initially met with skepticism, his findings were later confirmed by other scientists. Antonie van Leeuwenhoek is rightfully recognized as the first true microbiologist. His work established microbiology as a scientific field and revealed an entirely new world of microscopic life. Over his 50-year scientific career, he made over 500 microscopes and numerous groundbreaking discoveries. For centuries, scientists debated whether life could spontaneously arise from non-living matter. The theory of spontaneous generation proposed that living organisms could arise directly from non-living matter. Common beliefs included that maggots emerged spontaneously from rotting meat, mice generated from stored grain, and frogs and insects formed from mud. Let's look at the historical context of this controversy. The concept originated with Aristotle in ancient Greece. For over 2,000 years, spontaneous generation was widely accepted by scholars and scientists. During the scientific revolution of the 1600s, new observational methods led some scientists to question this long-held belief. In 1668, Francesco Redi conducted the first controlled experiments challenging spontaneous generation, which we'll explore in the next section. Throughout the 1700s, the debate intensified as scientists on both sides performed various experiments. Finally, in 1859, Louis Pasteur's elegant experiments definitively disproved spontaneous generation. The controversy sparked intense scientific debate. Supporters of spontaneous generation cited everyday observations that seemed to confirm the theory. They also noted the lack of alternative explanations for life's appearance and relied on centuries of scholarly tradition. Opponents argued that all life comes from existing life. They conducted controlled experiments showing different results and used improved microscopes to reveal previously unseen organisms. The resolution of this controversy established a cornerstone principle of modern biology: all life comes from existing life. This scientific breakthrough demonstrated the importance of controlled experiments, led to advances in sterilization techniques, and paved the way for the germ theory of disease. In the next section, we'll explore how Francesco Redi conducted the first scientific experiments challenging spontaneous generation. In 1668, Italian physician Francesco Redi conducted one of the first controlled experiments challenging the prevailing theory of spontaneous generation. Redi designed a simple but elegant experiment. He placed pieces of meat in 3 identical jars with different coverings. One jar was left completely open to the air. A second jar was covered with fine gauze, allowing air to enter but preventing flies, and a third jar was completely sealed. After several days, Redi observed that maggots appeared only in the open jar where flies could freely enter and lay eggs. No maggots appeared in the gauze-covered or sealed jars. Redi's findings were significant for several reasons. He demonstrated that at least some organisms, like maggots, don't spontaneously generate but require parent organisms. His experiment introduced controlled variables and the scientific method to biology, comparing identical conditions with one key difference. However, Redi's experiment had limitations. It didn't address microscopic life, which hadn't been well studied yet. Some still claimed that microorganisms could appear spontaneously. While Redi's experiment was groundbreaking, the debate over spontaneous generation would continue for nearly 200 years, with scientists like Spallanzani and eventually Pasteur building upon his methodology. Lazzaro Spallanzani, an Italian priest and biologist, conducted pivotal experiments challenging spontaneous generation in the 1760s. Spallanzani designed an elegant experiment to test whether microorganisms appeared spontaneously or came from the air. He first boiled meat broth in both flasks to kill any existing microorganisms. After sealing one flask while leaving the other open, Spallanzani observed that microorganisms appeared only in the open flask. Critics of Spallanzani's work argued that he had excluded vital air, what we now call oxygen, which they believed was necessary for spontaneous generation to occur. Despite these criticisms, Spallanzani's meticulous experiments represented significant progress in applying the scientific method to microbiology. His work influenced later scientists like Louis Pasteur, who would definitively settle the spontaneous generation debate. Louis Pasteur, a French chemist who lived from 1822 to 1895, made crucial discoveries that would forever change our understanding of microbiology. In the mid-19th century, many still believed in spontaneous generation, the idea that living organisms could arise from non-living matter without any parent organism. In 1861, Pasteur designed an elegant experiment to definitively resolve this long-standing controversy. Pasteur's famous experiment used specially designed swan neck flasks. he first prepared 3 types of flasks containing nutrient broth. He boiled the broth in all flasks to kill any existing microorganisms. This sterilization process was a critical step in the experiment. In all flasks, Pasteur allowed air to enter, but in the swan neck design, the curved neck acted as a trap for airborne microorganisms in dust particles. The results were clear. The broth in regular flasks and those with broken swan necks became contaminated with microbes, but the intact swan neck flasks remained sterile indefinitely. This elegantly designed experiment conclusively disproved spontaneous generation and established the principle of biogenesis, that living organisms arise only from pre-existing living organisms. Pasteur's rigorous experimental approach not only resolved a long-standing scientific debate, but also laid crucial groundwork for the development of modern microbiology, sterilization techniques, and germ theory. This landmark achievement was just one of Pasteur's many contributions to science and medicine. Louis Pasteur made groundbreaking discoveries about fermentation in the 1850s and 1860s. Prior to Pasteur's work, fermentation was thought to be a purely chemical process. Through careful experimentation, Pasteur demonstrated that it was actually caused by living microorganisms. Pasteur identified yeast cells as the microorganisms responsible for alcohol fermentation in beer and wine. Pasteur's research led to the development of pasteurization, a process that uses controlled heat to kill harmful microorganisms in liquids. The process involves heating the liquid to a specific temperature, maintaining it for a set time, and then rapidly cooling it. Pasteur initially developed pasteurization to prevent wine and beer from spoiling. Later, the process was applied to milk and other food products, dramatically reducing foodborne illness worldwide. Pasteur's development of pasteurization revolutionized food safety worldwide and has saved countless lives by preventing the spread of harmful bacteria through food and beverages. Pasteur's work on fermentation and pasteurization remains one of the most significant contributions to food science and public health in history. The germ theory represents a revolutionary understanding in medical science. At its core, the germ theory established that specific microorganisms cause specific diseases. This fundamental concept is considered one of the most important developments in the entire history of medicine. The development of the germ theory was primarily the combined work of several 19th century scientists. Louis Pasteur made crucial contributions by disproving spontaneous generation and showing that microbes cause fermentation. Robert Koch identified specific bacteria as the causative agents of diseases like anthrax, tuberculosis, and cholera. The germ theory represented a complete paradigm shift in how medicine understood and approached disease. Before the germ theory, diseases were thought to be caused by bad air, imbalance of bodily humors, or divine punishment. After its establishment, medicine recognized that specific pathogens cause diseases, leading to new approaches focused on prevention, hygiene, and scientific treatment. This fundamental shift in understanding disease causation formed the foundation for modern medicine and public health practices that we rely on today. Robert Koch, a German physician from 1843 to 1910, made monumental contributions to the field of microbiology. In 1876, Koch identified Bacillus anthracis as the cause of anthrax, providing the first definitive proof of the germ theory of disease. His methodical approach established a systematic way to prove that specific bacteria cause specific diseases. One of Koch's most significant contributions was the development of solid culture media, initially using gelatin and later agar. This innovation allowed scientists to isolate pure bacterial cultures, a technique that remains fundamental to microbiology laboratories today. Let's look at the timeline of Koch's life and major achievements. Born in 1843, Koch made his breakthrough with anthrax in 1876. Through the 1880s, he refined his methods and discovered the bacteria responsible for tuberculosis and cholera. In 1905, Koch was awarded the Nobel Prize in Physiology or Medicine for his investigations and discoveries in relation to tuberculosis. After his death in 1910, his legacy continued through his methodologies that revolutionized how we study and understand infectious diseases. Koch's scientific legacy extends beyond his specific discoveries. His systematic approach to disease causation formed the foundation for Koch's postulates, which we'll explore in the next section. Koch once said, "Science knows no country because knowledge belongs to humanity and is the torch which illuminates the world." His meticulous methods and culture techniques transformed microbiology from observation to experimental science, paving the way for countless medical advances. Robert Koch, a German physician and microbiologist, established a groundbreaking methodology in the 1880s to prove that specific microorganisms cause specific diseases. Koch established 4 criteria, now known as Koch's postulates, that must be satisfied to prove a specific microorganism causes a specific disease. Postulate 1. The microorganism must be present in all cases of the disease. This means researchers must find the same microbe in every infected individual, but not in healthy ones. Postulate 2. The microorganism must be isolated and grown in pure culture. Scientists must extract the suspected pathogen and grow it separately from all other organisms. Postulate 3. When the pure culture is introduced into a healthy, susceptible host, it must cause the same disease. This step confirms the microorganism's pathogenic capabilities. Postulate 4. The same microorganism must be re-isolated from the experimentally infected host and identified as being identical to the original specific causative agent. This final step closes the loop of proof. Koch's postulates became the gold standard for proving disease causation in microbiology. They provided a methodical framework that revolutionized the scientific understanding of infectious diseases. While still fundamental to microbiology today, these postulates have known limitations when applied to viruses, polymicrobial diseases, or microorganisms that cannot be cultured in the laboratory. The period from approximately 1875 to 1915 is often called microbiology's golden age. During this remarkably productive era, scientists identified the causative agents of many major diseases, transforming medicine from a largely symptomatic practice to one based on specific disease causation. Let's explore the major discoveries during this golden period of microbiology that spanned 4 decades. In 1882, Robert Koch identified Mycobacterium tuberculosis, the bacterium causing tuberculosis, a disease that had plagued humanity for centuries. In 1883, Koch discovered Vibrio cholerae, the causative agent of cholera, during an epidemic in Egypt and India. Klebs and Loeffler identified Corynebacterium diphtheriae in 1884, the bacterium responsible for diphtheria, a major cause of childhood mortality. Alexandra Yersin discovered Yersinia pestis, the bacterium causing plague, during an epidemic in Hong Kong in 1894. The causative agent of syphilis, Treponema pallidum, was identified by Schouten and Hoffman in 1905. Shiga discovered the bacterium causing dysentery in 1898, which was later named Shigella dysenteria in his honor. These discoveries fundamentally transformed medicine from a largely symptomatic practice to one based on specific disease causation. The identification of specific pathogens enabled targeted treatments and preventive measures. Diagnostic techniques were developed to identify these pathogens. This laid the foundation for vaccines and other preventive approaches, while establishing protocols for public health that would save countless lives. The golden age of microbiology established the field as a cornerstone of modern medicine, setting the stage for future advances in immunology, virology, and eventually molecular biology. The study of the body's defense against microbes developed alongside microbiology, establishing what we now know as immunology. Let's explore the key developments in early immunology from 1796 to 1908. Edward Jenner made the first immunological breakthrough in 1796. He discovered that inoculation with cowpox protected against smallpox, creating the first vaccine, though the mechanism wasn't understood at the time. This discovery was revolutionary, though it would take decades to understand how vaccines actually worked. Louis Pasteur advanced immunology by developing attenuated vaccines. weakened versions of disease organisms. Between 1879 and 1885, Pasteur created vaccines for chicken cholera, anthrax, and notably rabies. Pasteur's method of attenuation became a cornerstone of vaccine development, but the cellular mechanisms of immunity remained unknown. In 1883, Elie Metchnikoff discovered phagocytosis, the process where specialized white blood cells engulf and destroy bacteria. This established cellular immunity as a key defense mechanism. Metchnikoff's work on cellular immunity would later earn him the Nobel Prize. In 1890, Paul Ehrlich proposed the side chain theory of antibody formation. He suggested that cells have receptors, or side chains, that bind to specific toxins. This established the foundation of humoral immunity, the antibody-mediated defense system. These pioneering discoveries, Jenner's vaccination, Pasteur's attenuated vaccines, Metchnikoff's cellular immunity, and Ehrlich's humoral immunity, established immunology as a distinct but related field to microbiology. Their work formed the basis for our modern understanding of how the body defends against disease. Paul Ehrlich, a brilliant German scientist who lived from 1854 to 1915, revolutionized medicine with a radical new idea. He pioneered the concept of chemotherapy, the idea that specific chemicals could be used to treat disease. As he famously stated, we must learn to aim chemically. Ehrlich envisioned what he called a magic bullet approach. He believed it was possible to develop chemicals that could selectively target disease-causing organisms while sparing the host's healthy tissues. Ehrlich's pursuit of this magic bullet was methodical and relentless. Beginning in 1905, he systematically created and tested hundreds of compounds. Finally, in 1909, after 605 unsuccessful attempts, his 606th compound, later named salvarsan, proved effective against syphilis. Salvarsan represented a watershed moment in medical history. It was the first effective treatment for syphilis, a devastating disease that had plagued humanity for centuries. More fundamentally, it was the world's first chemotherapeutic agent, proving that chemicals could be designed to fight disease. Ehrlich's work established the principle of selective toxicity, that drugs could be created to harm pathogens while leaving human cells intact. Ehrlich's methodical approach laid the foundation for modern pharmaceutical research and development. His work paved the way for the development of antibiotics in the following decades. And his magic bullet concept continues to inspire modern targeted therapies, including those used in cancer treatment today. In 1928, Alexander Fleming made a groundbreaking discovery that would forever change medicine. While studying Staphylococcus bacteria, Fleming noticed something unusual in one of his petri dishes. A mold called penicillium had contaminated his culture, and surprisingly, no bacteria were growing around it. Fleming observed that the mold was producing a substance that inhibited bacterial growth. He named this substance penicillin. Despite this discovery, it took over a decade before penicillin would be developed for medical use. In the early 1940s, Howard Florey and Ernst Chain led a team at Oxford University that finally developed methods to isolate and purify penicillin. They developed techniques to purify penicillin and demonstrated its remarkable effectiveness against many bacterial infections. The timing of this breakthrough was crucial, as World War II was raging. Penicillin became vital for treating wounded soldiers. With government support, pharmaceutical companies rapidly scaled up production. By 1945, penicillin was widely available to Allied troops, saving countless lives from infections. The success of penicillin triggered what we now call the antibiotic era, a period of rapid discovery and development of new antibiotics. Throughout the 1950s and beyond, scientists discovered numerous new antibiotics, each targeting different types of bacteria. The impact on public health was dramatic. Mortality rates from bacterial infections plummeted, transforming once deadly diseases into treatable conditions. Virology emerged as a distinct branch of microbiology in the late 19th century. In 1892, Dmitry Ivanovsky discovered what would later be identified as the tobacco mosaic virus. This was an infectious agent that could pass through filters that trapped bacteria. This discovery challenged existing understanding of pathogens. In 1898, Martinus Beijerinck expanded on this work. He termed this mysterious pathogen a contagium vivum fluidum, meaning contagious living fluid. This conceptual breakthrough would await technological advancement to be fully understood. It wasn't until the invention of the electron microscope in the 1930s that scientists could finally visualize viruses. This technological breakthrough led to rapid advances in understanding these unique microorganisms. The mid-20th century witnessed a revolutionary merger of microbiology with the emerging field of molecular biology. This convergence of scientific disciplines revolutionized our understanding of life at its most fundamental level. Experiments with bacteria and bacteriophages, viruses that infect bacteria, became crucial tools for revealing fundamental genetic principles. In 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty conducted a groundbreaking experiment with pneumococcus bacteria. They extracted DNA from virulent smooth colony bacteria and demonstrated that this DNA alone could transform non-virulent rough colony bacteria into the virulent smooth form. This landmark experiment provided the first conclusive evidence that DNA, not proteins, was the molecule responsible for heredity. Building on X-ray crystallography work by Rosalind Franklin and Maurice Wilkins, James Watson and Francis Crick published their landmark double helix model of DNA structure in 1953. The double helix with complementary base pairing explained how genetic information could be stored and accurately replicated through semi-conservative replication. Following the DNA structure discovery, bacteria became essential model organisms for understanding fundamental biological processes. Using bacterial systems, scientists deciphered gene regulation mechanisms, protein synthesis pathways, and the genetic code itself. François Jacob and Jacques Monod's work on the lock operon in E. coli revealed how genes are turned on and off, a discovery that earned them the Nobel Prize and revolutionized our understanding of gene regulation. The mid-20th century convergence of microbiology and molecular biology fundamentally transformed our understanding of genetics and heredity. From Avery's demonstration of DNA as genetic material in 1944, through the Watson and Crick model in 1953, to the development of recombinant DNA technologies in the 1970s, microbial systems proved invaluable for revealing life's molecular foundations. These discoveries laid the groundwork for modern genetic engineering, biotechnology, and genomics, forever changing medicine, agriculture, and our fundamental understanding of life itself. By the mid-20th century, scientists began to develop a deeper appreciation for the crucial roles microorganisms play in ecology and the environment. Two significant pioneers in this field were Sergei Winogradsky and Martinus Beijerinck, who laid the foundations for environmental microbiology. Scientists discovered that microbes perform essential roles in nutrient cycling, including carbon, nitrogen, and phosphorus cycles. They contribute significantly to soil fertility by decomposing organic matter and releasing nutrients that plants can use. Microbes are also fundamental to overall ecosystem health, playing roles in pollution remediation and forming symbiotic relationships with plants and animals. This expanded understanding transformed microbiology from a primarily medical discipline focused on disease-causing agents. Microbes came to be recognized as fundamental components of all ecosystems and essential to Earth's biological processes. This fundamental shift in perspective established environmental microbiology as a crucial scientific discipline, recognizing microbes as the essential architects of our environment, not just agents of disease. This is how microbiology transformed public health practices across the world. Microbiology profoundly impacted public health practices. Understanding disease transmission led to improved sanitation, water treatment, food safety measures, and the development of vaccines. Understanding waterborne pathogens through microscopy led to revolutionary water treatment systems. John Snow's landmark cholera investigation in 1854 demonstrated the link between contaminated water and disease. By the early 1900s, water filtration and chlorination dramatically reduced waterborne illnesses. Vaccines developed through microbiological research dramatically reduced the global burden of infectious diseases. Beginning with Jenner's smallpox vaccine in 1796, the scientific understanding of immunity led to life-saving immunizations against many deadly pathogens. This progress culminated in the complete eradication of smallpox by 1980, one of public health's greatest achievements. The establishment of public health laboratories in the late 19th and early 20th centuries created critical infrastructure for disease surveillance and outbreak investigation. These specialized facilities developed the capability to identify and track specific pathogens, enabling rapid response to emerging threats. The 1993 Milwaukee cryptosporidium outbreak demonstrates how laboratory detection capabilities can identify waterborne pathogens and protect communities. Microbiological research transformed food safety practices. The development of testing methods to identify specific pathogens like Salmonella and E. coli allowed for targeted interventions. Process controls such as pasteurization, developed by Louis Pasteur in the 1860s, and modern hazard analysis critical control point systems significantly reduced foodborne illness rates across developed nations. The impact of microbiology on public health has been transformative. Life expectancy increased from 30 to 40 years in 1900 to over 70 to 80 years today, while child mortality decreased from nearly 30% to less than 1% in developed nations. These applications of microbiological knowledge have saved countless lives and remain central to protecting population health in the modern world. Microbiology continues to be a cornerstone of modern public health practices worldwide. The late 20th century brought revolutionary molecular techniques that transformed how we study and understand microorganisms. Three key molecular techniques fundamentally changed microbiology: polymerase chain reaction, DNA sequencing, and genetic engineering. Polymerase chain reaction, or PCR, allows scientists to make millions of copies of a specific DNA segment in just a few hours. DNA sequencing determines the exact order of nucleotides in a DNA molecule, revealing the genetic information encoded within. Genetic engineering provides tools to modify microbial DNA, allowing scientists to alter their properties or make them produce useful compounds. PCR works through repeated cycles of temperature changes. First, DNA is denatured at high temperature, then primers attach at lower temperatures, and finally, DNA polymerase extends the new strands. These molecular techniques revolutionized microbiology by enabling identification without cultivation, rapid pathogen detection, and manipulation of microbial genetics. Molecular techniques in microbiology have continuously evolved since the discovery of DNA's structure in 1953, with each advancement bringing new capabilities to microbiologists. These techniques continue to evolve, enabling increasingly sophisticated understanding of the microbial world and opening new frontiers in research and applications. The future of microbiology represents a rapidly evolving frontier of scientific discovery. Synthetic biology aims to engineer microbes with specific functions for medical treatments, industrial processes, and environmental remediation. Metagenomics allows scientists to study entire microbial communities without culturing individual species, revolutionizing our understanding of microbial ecosystems. Microbiome research explores the complex relationships between humans and their microbial inhabitants, opening new avenues for treating various conditions from digestive disorders to mental health. One of the greatest challenges facing modern microbiology is antimicrobial resistance, which threatens to undermine decades of progress in treating infectious diseases. The emergence of new infectious diseases, accelerated by climate change and globalization, ensures that microbiology remains critically important to global public health. Advanced technologies like CRISPR gene editing, nanopore sequencing, Artificial intelligence, biocomputing, and single-cell analysis are transforming microbiological research. The historical journey of microbiology continues, building on centuries of discovery while embracing new frontiers of scientific exploration. As we face global challenges, microbiology remains at the forefront of science, offering solutions for human health and environmental sustainability.